Thanks for the opportunity to speak today. So what I'd like to talk to you about is the project that we had funded through NESP Tropical Water Quality Hub starting just in January this year. And this project is focused on understanding how water quality might impact bleaching susceptibility and also recovery from bleaching. So I don't have to educate this audience that extreme temperatures or marine heat waves are incredibly detrimental to corals, and they will bleach when they experience these more extreme temperatures. We've seen mass bleaching on the Great Barrier Reef 4 times since 1998, and most significantly twice in the last 2 years. So we've just seen consecutive bleaching events. There's actually no evidence of bleaching going back in records of coral cores beyond 1998. So this is really a very modern phenomenon and something that we should all be very concerned about. So here's a photograph or an image that was published in The Guardian, where the results of the aerial surveys conducted in 2016 and 17 are displayed. And what you can see is that in 2016, the bleaching was very much focused in the top third of the Great Barrier Reef, whereas in 2017, the impact was felt more strongly further to the south in the central sector, just off the coast of Townsville and extending up just beyond Cairns. And while the mortality estimates are still being refined and put together, it appears that at least the 2016 event, for which the numbers are much firmer at this point in time, resulted in quite extensive mortality, particularly in the far northern sector of the Great Barrier Reef, with up to 83% of coral cover declining in these, or 83% of corals dying in these regions. Overall, the mortality is estimated at about 30% across the Great Barrier Reef. And here's a couple of images that we took while we were out there surveying just this year. So here's a photograph of a reef near Cairns at the height of bleaching, and you can see all the very white colonies with mortality starting here and mortality in the foreground here. Here's another photograph actually taken by Rachel down the back from off Mission Beach, and again you can see that the branching corals are heavily bleached. Some of them are still retaining some fluorescent colour, but this is a part of the bleaching process. So our project within the NESP funding scheme is to try and understand what the linkages are between water quality and bleaching. And our project has 3 parts. So we do surveys, so we record the bleaching in the field across water quality gradients in the central sector, and we've done this for 2 years, in 16, and we repeated them in 17 when the reef bleached again. We have a large component that's aimed at understanding the physiology of bleaching and how water quality can impact that process. And then lastly, we work with the modelers at CSIRO to use the e-reefs model to generalise our results and to understand how water quality improvements can actually mitigate some of these impacts that we're expecting from global warming and that we're expecting to increase over time. So here's the team: Mark, Neil Canton, Katerina, Kate Quigley, a PhD student, and also we're working with Catherine Martin and Rachel Pears from the Marine Park Authority. And the questions that we're asking are, what are the correlations of water quality with bleaching and the heat experienced on the reef? We're aiming to understand what are the most important water quality parameters, and also to identify the temperature range over which water quality potentially makes a real impact or makes a real difference as to how much corals will bleach. We are also interested in understanding the physiological and genetic mechanisms underpinning bleaching susceptibility, in particular whether corals have mechanisms that allow them to combat the stress that they're experiencing and the role of the algal symbiont. And then we aim to use this information to generalise to the reef more broadly, and to understand what end-of-river load improvements may affect bleaching susceptibility. So before I go on to talk about some of our early results, I just want to emphasise that corals live in symbiosis with microbes. They are associated both with bacteria, viruses and fungi, but also importantly with the algal symbionts that are lost during bleaching. The types and the relative abundance of these microbes are essential for health. And they may be locally adapted, such that there is an optimal community and an optimal abundance of microbes under local conditions. And these may vary across environmental gradients. So if we imagine the environmental gradient down here as a spectrum, we might get the optimal health and performance of a particular microbial community, but for that may actually vary across environmental gradients. So how does bleaching occur? Well, firstly, we have the coral holobiont. Here's a photograph of a coral with the symbionts in the tissues here in green. And what happens is that we have environmental conditions that go out of the ordinary, and this can result in a disruption of the photosynthetic apparatus. What happens is that there will be the algae will produce increased amounts of reactive oxygen species, which are toxic. And if, for example, we're talking about extreme heat, the proteins in cells might actually start to denature. This results in cellular stress, then a decrease in the algal cells follow. This is the bleaching process. But there are things that corals can do to combat that. They can use antioxidant and their immunity response to ameliorate at least some of the early effects of the stress. And there are genetic factors that can impact this, not only from the coral host, but also from the symbionts. And what we've learnt is that although the algal symbionts look very similar, they are in fact quite genetically diverse. And certain types, there are about 9 major types recognised at this point in time. Some of them are more tolerant than others. So for example, this D type tends to be more tolerant, and corals that host the D type will experience less stress given the same amount of warming and light than others hosting, for example, C types. So once the bleaching is experienced, then there's 2 outcomes: either the coral dies or it recovers. And our project is very focused on understanding why some will die and others recover. So what are the interactions? How can water quality actually interact with this process, and how may it influence bleaching susceptibility? There's quite a few different mechanisms proposed, and they are rather complicated, but I'll just try and give you a broad overview. So there's a number of hypotheses that relate to nitrogen enrichment. So we have enhanced nitrogen in the environment that stimulates zooxanthellae growth, so we get an elevation of the zooxanthellae populations, and this leads to a nutrient limitation either of phosphorus or perhaps also carbon, and this results in poor photosynthetic health or poor cellular health, and so this can exacerbate bleaching. So essentially we have too much nitrogen. As the algal cells grow, they run out of phosphorus and then they become less healthy. And there's experimental evidence for this. Another mechanism relates to carbon enrichment. So you can have an enhancement of carbon in reef waters, either through water quality issues, but also through the organisms excreting carbons themselves. Again, this stimulates the growth of nitrogen-fixing bacteria, and we end up in the same loop where there's an elevation of nitrogen, which leads to a decrease in the phosphorus and poor cellular health. And there are some experimental tests of this hypothesis as well. Lastly, turbidity is a very important factor, water quality factor impacting coral health. And it can affect both the available light but also the variation in light. So when we see bleaching conditions, it's often following a heat wave where the water, where the air is still, there's very little wind, and during these conditions the turbidity can decline so that the water becomes clearer and there's a large change in the light available for corals. So if they're adapted to dimmer conditions, they're actually experiencing a lot extra light than what they would normally see. But turbidity is not always bad. It can support food webs that feed corals. Here's an example of a paper we had published earlier this year where we found that there was large variation in growth, for example, of corals across water quality gradients. So turbidity can also impact the food webs, and corals that are fed tend to bleach less or recover sooner as well. So again, these are quite complicated and complex interactions. Nitrogen-rich particles are not always good though. They can lead to enhanced bleaching as well. So we have been studying the marine heat waves over the past 2 years that have hit the Great Barrier Reef. And the way that we look at the amount of heat that the corals experienced is through the NOAA degree heating weeks metric. So this is a metric that quantifies the amount of additional heat that corals are experiencing in relation to their maximum summer temperatures. And you can visualize this on a scale from 0 degree heating weeks, meaning that they haven't experienced heat stress. 1 degree heating week is 1 week spent above this maximum summer temperature, and so on. Normally we saw degree heating weeks of 4 to be the level where corals bleach, and about 8 being the level where they die. But actually following these last 2 events, those numbers have been downscaled a little bit, and actually bleaching is observed at about 2 degree heating weeks and mortality at about 6. So here's the heating that was observed on the Great Barrier Reef over these 2 events. So you can see once we're getting between the yellow and red colours, it's kind of bad, and a lot of the Great Barrier Reef, particularly the northern part, experienced that in 2016, and in 17 we saw the central part much warmer and the bleaching as predicted based on this heating. So we've been surveying about 20 reefs across water quality gradients. We've reduced that down to 14, and these maps are probably a little bit hard to see from the back, but you can see here in the different colours is the severity of bleaching observed on that reef, with red being particularly bad. So in 16, the bleaching wasn't too bad in the central sector. It was really up in the northern part that we started to see severely bleached reefs. But in 17, the picture is much different, where you can see many more red reefs in the central sector here, and you can see the paired design of inshore versus offshore comparisons to give us more power to understand how water quality might impact whether a reef is blue or red. And we actually recorded quite differential bleaching responses on the reefs that we went to. So as predicted, bleaching increases with heat stress. That we would have been surprised if we hadn't seen that. What I've plotted here is the percentage of colonies or data points that were in the severe bleaching or mortality category. So these are corals that are really white and sort of showing starting mortality. So these are on the way out. And what you can see here is 16 in one panel and 2017 in the other panel. That all the reefs that we visited and all the transects that we did in 17 were in the severe or the early mortality category. But what I want to highlight to you is that along the x-axis here is the heat exposure or the degree heating weeks. So you can see at a set degree heating week, we're actually experiencing a lot of variation. So there's variation in how badly bleached reefs are given a similar heat exposure. And that's really what our project is trying to tease out, the role that water quality can play in producing this variability. So, as I mentioned, we're coming to the end of our first year. We have been quite busy, but there's still a lot remaining to do. And some of the questions that we're going to be addressing in the second year of the project are better understanding how physiological stress and the symbiosis how the samples we've collected from the field, what they can tell us about physiological stress and symbiosis.